Transducer, we have seen that active and passive. It is now first passive. Here's the passive transport, and in case of passive transport, it is the passive diffusion. Passive diffusion. Okay. As I have told you that passive diffusion is mainly based on the Fick's law. Fick's first law. It is written here that Fick's first law. It is the dq by dt is the rate of transport is directly proportional to diffusion constant, area, and partition coefficient of the drug molecule, and the thickness of the membrane. Thickness of the membrane. Here it is the CGIT, it's the concentration of the drug available at site of action, and C, this refers to the, when we consider, this one is the biological membrane, and this is the biological membrane, this is the part of GIT, and this is the, suppose this is the blood. Then this CGIT lies here, and C will lie here. So here you can see that the drug molecule will be transported here and concentration will gradually increase. So here in this process, generally this rate is mainly dependent on the area available, and as we all know that this is the constant, this one is also constant, this thickness will be also constant, but area available, which is the major factor. Generally when the drug molecule, it is unionized in particular portion of the GIT, because ionization pK and the pH, these three have the correlation between each other. So drug molecule which is having the particular pK value will be ionized at on the particular part of GIT only, and the availability of area at on that part of GI tract will affect the absorption of the drug, means the transport of the drug to the biological membrane. Okay, so area is given the main consideration here, because these three are the constant, and another one is the concentration of drug available at the site of action. Now this process is termed as the downhill process and non-ionic, because ionized molecule cannot pass through the membrane easily because of the structure of the membrane, which allows the passing of only unionized molecule, lipophilic unionized molecules. In case of downhill process, why it is termed as a downhill process? Generally, you can see this one is a slope. If I have to go through this side to this side, you just think about you are going through the steps. Okay, when you are going upward, you have to give off efforts. But when you are coming down, it is very easy for you to come down. So here it is something like coming down. It is really very easy. So this one is termed as a downhill process. When in case of active transport, where energy is required, it is termed as the uphill process. Okay. So here is the downhill process. Non-ionic molecule will pass through it, and it is the non-saturable. In case of active transport, you know that some carrier molecules are there, so it will be saturable and it will follow the mixed order kinetics, while here it only follows first order kinetics, and the limit of the molecule which can pass through is the 100 to 400 Dalton. However, cyclosporine like drug which having the molecular weight of 1200 Daltons can easily pass. There are some exceptions are there. Sink condition. We all know in case of blood, plasma proteins will be there. Plasma proteins are present in the blood compartment. So whenever our drug passes through the membrane, this is the passage of molecule through the membrane, directly this drug molecule will bind to this plasma protein according to its affinity. And mainly the affinity of the drug molecule to the plasma protein is based on its lipophilicity. Higher the lipophilicity, higher will be the affinity to the toward the plasma proteins. So here drug molecule will bind to plasma protein, and due to this process continuous sink condition will be maintained, and due to this sink condition I can consider that this part is continuously zero. So when I consider this part continuously zero, then equation becomes like this, that the rate of diffusion is directly proportional to dAk by h Cgit. If I consider this whole thing as one constant, and I say this constant as the permeability P, because this all parameters are based on permeability. When you go for partition coefficient, it is based on permeability. Area, it is also there, and diffusion is also based on permeability, and thickness is also based on permeability. So when I take this one as a P, as a permeability constant, okay, so I can rewrite this equation like this: dq by dt is equal to P and concentration Git. So here's the equation comes, which shows that the rate is following the first order kinetics, where it is the constant and it is following the first order. So it is dependent on the concentration present at site of action, and this is all about the passive diffusion. Now next topic, intensive wheel. Under the passive, it's the pore transport and ion pair transport. Just two other small topics are there. First, the pore transport. It is the, you can say it's a convective transport, bulk transport, or generally a simple filtration. Why filtration? Because in this case, what we are watching in this filtration, that solvent will pass through the certain membrane, and then only the liquid material will come out. And same things happen here. But suppose you think that the filter is having some impurity, or maybe some cracks are there. So with the solvent, certain parts of filter media or some molecules from the upper cake part will come out. So same thing will happen here. That this one is the biological membrane, and when your molecule, suppose your molecule is here, it is the so small molecule, and with the solvent drag, suppose this is the drag which is due to the solvent, with this drag your molecule also passes through the membrane. You all know the osmosis. Osmosis is the, you can say, the hydrodynamic pressure or osmotic pressure. The osmotic pressure, in case of osmosis, what happens to the semi-permeable membrane? We all know that biological membrane is a semi-permeable membrane. Through this semi-permeable membrane, the the solvent system will pass. Solvent will pass through the membrane, and generally we know that only solvent will pass. But sometimes what happens, these small molecules can also pass through with the solvent. So same thing happens here, that due to hydrodynamic pressure, the small molecule like which is having the weight about 100 Dalton can pass through the membrane. And in certain cases, when the molecule is there as a linear chain, okay, so linear chain can also pass by means of this transport mechanism. However, it is having the molecular weight up to 400 Dalton. And what is the importance of the pore transport? This is pore transport. We all know the major three organs: it's the liver, brain, and the kidney. In these all organs, mainly if the accumulation of drug takes place, it may create some problems. So in this pore transport mainly helps in the excretion of the molecules from this three part: brain, liver, and kidney. Okay. Now we'll move toward the ion pair transport. We all know that pH-pK relationship is mainly based that the ionization, how the ionization of the molecule takes place in GI milieu. In general, if I consider that in case of stomach, it's suppose pH is about 2. pH is 2 in stomach, and my molecule which is having the pK value of 4. So here what happens, that molecule which is having a pK value of 4 will be 100%. According to the Henderson-Hasselbalch equation, it will be the 99.99% unionized form. Okay. So due to this 99% of unionized form, we all know that it will not able to solubilize, and solubility is also one of the requirement beside the unionization. But here, when suppose this pH changes, with changing, And if now we think about the fed state, in which the stomach pH will raise up to the 5. In case of fed state, stomach pH will generally raise out 4 to 5. If I consider 5, then what happens? Your molecule now in such condition where about 50 percentage, now slightly more than 50%, you can consider, suppose the 70 percentage is the approximate value. Suppose 70 percentage is in ionized form and 30 percentage will be unionized. So now this unionized, which is the sum having the some solubility, will go for the transport. But when this 70 percentage, which is ionized, still what happens? The drug which is having the absorption window in stomach and taken after the food. So what happens? This 70 percentage drug, what happens to it? Sometimes this transport mechanism, it helps in it. How it helps? That suppose we consider some endogenous moiety. We will consider mucin. Mucin is generally a negatively charged protein. You can say protein molecule which is negatively charged. Okay. So when we are having the drug which is having the positive charge, how this positive charge generated, that I have explained, that by means of pH-pK relationship, this charge will generate due to the ionization. So when it is having the positive charge, or mucin moiety will be having negative charge, and simple neutralization, you cannot say the neutralization, but you can say it will generate the molecule which is having no charge. This molecule will not have any charge. So what happens? Now this, once the endogenous moiety bound to the above drug molecule which is ionized, it forms the molecule which is unionized. And now this molecule will pass through the membrane by means of simple passive diffusion. This is termed as the ion pair transport.